Thank you everyone for being here in the image analysis session. We will be sharing the session, Pedro, Pereira, and myself and Arrate Muñoz. And I'm delighted to introduce to you Martin Weigert, who is a computer scientist who research focus on computational optics and machine learning, based image reconstruction and segmentation for microscopy. He did his PhD and the MPI CBG in Dresden, Germany, and recently he joined EPFL in Lausanne, Switzerland, as a group leader. Please, Martin, when you like.
Thank you, Arrate. Can you see my screen?
Yes, we can.
Okay, perfect. Then thank you very much for the nice introduction, and welcome everyone to my talk titled Nuclei Detection and Segmentation with Learned Shape Representations. Very thankful for the organizer for inviting me. First time Baum, so I'm very excited to be here and speak a little bit about my research. As Arrate already mentioned, I recently joined EPFL Lausanne as a junior group leader in a perfect timing of a pandemic. And I will give you a rough overview of what I've done so far and what I will do next, and specifically focus on nuclei detection and segmentation. So this is just a short overview of my research interests and the things that we've done in the past and will do in the future. The first part is machine learning-based image processing. So this confines the restoration of clean signals from corrupted input data, such as denoising or deconvolution, and all with a focus on machine learning aspects, so pattern recognition or learning from in a data-driven way, what can be subsumed as what we call content-aware image restoration. The second part is computational microscopy. So this is a simulation of how the light propagates through the sample. It's about simulating adaptive optics approaches or wavefront estimation from images, again, in a fast and reliable way. And the third part is segmentation, and this calls for the quantitative extraction of semantically rich shapes from microscopy images where cells are crowded or very, very noisy images, or typically very challenging. And just to show you some of the images that I'm always fascinated by the richness of biological images coming from a variety of modern microscopy modalities. And this is just a short overview of what I always find super interesting just to look at. So this can be 2D fluorescence microscopy. Here's images of cells. This entails as well histopathology, where you have RGB images of whole slide images that can span several centimeters, where you can have a picture of cells at a single cell level. This can be bright field. Here is shown some stem cells developing in a culture dish. This can be as well 3D plus time volumetric microscopy, for instance, light sheet microscopy. This is like a C. elegans developing over time, or a fruit fly on the right from the Keller and Royer lab. Very extremely fascinating just to see the movement of cells over time. And then again, there are different modalities such as electron microscopy. This is a stack, a 3D volumetric acquisition with focused ion beam scanning electron microscopy from Andreas Müller and Harald Ress from Genelia. And all these images contain such a richness of information that it's always just a joy to just look at them. But they all come with a kind of slightly problematic aspect to it, especially when it comes to live cell images, which probably everyone is aware of, that there are inherent trade-offs in microscopy that you have to obey. And this is always, or most often, due that you want to ensure your sample health, and this limits your photon budget. I'm pretty sure everyone is aware with that, but I just want to mention it briefly, that this limited photon budget kind of dictates a certain quantitative or qualitative or dictates certain limits to what you can achieve in terms of the image quality that you get out. And this is confined to light exposure, where you are often limited by phototoxicity, resulting in noisy, corrupted images that you want to recover. This can be as well speed, where you're limited by motion blur, or a limited amount of Z spacing. If you want to do volumetric acquisitions, this is typically done with with more spacing in Z, so less resolution in the axial dimension along the optical axis. And this confines to the inherent limit of the spatial resolution of a microscope that is given by the cutoff in the Fourier space. And this all results in images that are less than perfect, that you aim to restore and recover the clean ground truth, so to speak, from with computational means to do any analysis afterwards. And that's what we set out like 2 years ago with some colleagues from Dresden, G. Myers, Schmidt, Le Croyer, and Florian Hugh. And we thought, okay, why we not just do it with a machine learning based approach that is very successful, has been very successful, being applied in the case of natural photographies in the computer vision community. And here the idea was pretty simple, and maybe many of you have already seen that, but I just want to remind you that the idea is extremely basic and very simple, right? You would just go to the microscope and acquire 2 different light conditions or stacks that are corresponding with 2 different light conditions. Here to the left, the light conditions that you want during your acquisition, and this is typically low signal noise ratio. And then you do with the same sample, just with a higher SNR, with a higher light camera exposure or laser power, the same stack at the same position, just with a higher SNR, and thereby generating perfectly well registered image pairs that you can then just train a deep regression model, or a regression model to start with, to recover the clean signal from the corrupted input image. And this regression model typically is just a deep convolutional neural network. And this is quite effective and works very, very well. And just to give you an example, this is a planarian flatworm that people wanted to image. And the problem here is that this planarian flatworm is extremely light sensitive. So the highest SNR that they could cope while imaging without the flatworm being just crawling out of their microscope was noisy images like this. And of course, you could acquire 2 different light conditions on fixed worms and then apply that trained network on living worms. And this is quite effective, as you can see. So most of the background and most of the corruption noise is actually removed from the sample. Thereby, any normal classical detection tool is capable of detecting way more cells than it could be in the original noisy images. Another example, which I found very illuminative, is the case of epithelial folding in Drosophila. Again, colleagues from Jason were interested in the shaping of the epithelial layer in the imaginal wing disc of Drosophila development. And this is an axial and XC slice of the raw images that they could acquire without corrupting or without compromising sample health. And they were interested in the geometric outline of this epithelial layer. So how it folds, what is the precise outline of these foldings, and what are tissue dynamics and the biophysical mechanisms resulting or are responsible being for this folding mechanism. And of course, like to be able to delineate this membrane very precisely, you need a certain amount of image quality, and this was just not present in the stacks they could acquire with at the normal light conditions. And here we used the same approach, just instead of denoising, we did additionally upsampling. So we acquire stacks with the normal typically conditions that is noisy and less well resolved along the axial dimension. And you can acquire corresponding stacks that have high laser power and are oversampled in Z. So they are really highly resolved in Z and a very high SNR. And you do the same trick and then apply it on live cell images. And as you can see, the restoration is so good that you can actually then follow those cells with with pretty much classical image analysis or segmentation tools, which you cannot do in the original stacks. And just to wrap up that shortly, that's a project that we implemented in Python, which is called CSPDeep. If you're interested in that, please go to this webpage or post questions on the forum. And this is like essentially a Python package that provides the backbone of our networks and the training and the model saving. So it's a nice flexible package. And most of the credit goes to Uwe Schmidt from Dresden, who is the strong working force behind all of that. And it's interesting in the sense that it not only provides code or provides the software that allows to train these kind of models, but it's kind of a very flexible platform to extend it even further. And I would mention some projects that kind of build on that and extend that in a very fascinating way. And you probably already heard this talk earlier in the conference, Zero Cost Deep Learning for Mike from the Henriquez lab. And maybe you have heard from Noise to Void from Kul and Florian Hug. And they all can leverage and tie onto that. And I think it's a very interesting way to share this, this code with everyone. Okay, coming to a little bit more semantic rich task, which is not only concerned with recovery of the image intensity, but actually getting meaning out of these images. And one of the most basal tasks that you can ask for, and which is often the first step in any bioimaging workflow, is the segmentation and detection of individual cell instances or nuclei from microscopy images. And this task can be actually categorized in two conceptually different ways you can do segmentation. So the input is always like an image that typically consists of a lot of crowded touching objects that contain a lot of out of focus areas. And the first thing you can do is just simply foreground background segmentation, and this is called dense segmentation. And this, of course, ignores that touching cells should be actually from a different class. And this is what is then called instant segmentation, where you not only want to do the foreground background, but you want to actually find out which of those different cell instances belong, or which pixels belong to different cell instances. So the task is given an input image. You want to find out and each ID that is specific to each cell. And what are common deep learning based approaches to do that? Okay, like typically this is a task prevalent in natural photographs too. So images of bikes or cars, if you ever see like a detectron architecture from Facebook that in real time, or from Tesla that in real time is able to segment objects, or like you probably have heard of the UNet that provides kind of a segmentation backbone too. And what are these typical approaches? That can actually be categorized in two things. The first approach is called, or I would call the bottom-up approach. This is you first do a foreground background segmentation, and then you try to discern different objects that are belong to different classes based on this foreground background first dense segmentation. And the second step is that it's called top-down. This is first you localize the images, and this is typically done with bounding boxes. So you first find for each object of interest, you find a bounding box, and then you try to refine this bounding box. For each instance that you found with such a bounding box, you try to precisely outline the shape within each bounding box. And this is an approach that works quite well in natural photography, but it has problems in microscopy. And I just want to mention there are other methods, for instance, affinity-based methods or embedding-based methods. For instance, a super cool latest paper by Carsten Stringer from Janelia Cell Pose that is a little bit different. But like the main two things are essentially these two, bottom-up and top-down. And the problems with microscopy images is that typically those microscopy images contain cells that are very crowded, very dense, and that are very, very blurry. So image boundaries are not very precisely outlined in the original image. And this leads to several problems. In the bottom-up approach, when you first do the foreground background segmentation, it tends to clump different distinct objects together in a single class. And it's hard. Typically what people do is do a watershed based on a distance transform, and that would fail in some cases, especially for crowded objects, which is quite prevalent in most bioimaging or a lot of bioimagical experiments. And the top-down approach has a different problem. And the problem is there that the primitive, the geometric primitive to detect cells is actually a bounding box that is ill-suited actually for roundish objects like cells, right? And if you look at this image here, for instance, to the right, you see that the bounding box of the center overlaps with the bounding boxes of the two neighboring cells. And typically top-down approaches would then just take or just leave the original, the center cell intact and discard the other two images because the bounding boxes are overlapping too much. And so we thought, okay, well, how can we do the top-down approach with bounding boxes, but without bounding boxes, with something that is more appropriate for cells? And so what we thought of, okay, instead of bounding boxes, we just predict more shapes, or shapes that are better representative of what happens or what you found typically for cell nuclei. And it's a very simple idea, but it works quite well. And this is, instead of a bounding box, you predict centers of nuclei. So you predict object probabilities, that is a peak at the center of each instance. And additionally, for every pixel, you predict the distances describing the circular outline around the center point. So it's kind of a polar coordinate representation for each pixel of the cell, or of the shape where it belongs to. And this can be predicted end to end. So you predict in the input images, is again an input. You would construct a network architecture that predicts both the object probabilities and the radial distances. And this is how it works. So you first do this dense polygon prediction. So for every pixel, you predict the object probability and the distance maps. And this you can typically just train normally with typically loss functions that are different for the probability and for the distances. And then when you have for every pixel, you essentially get for every pixel, you get the object probability and an outline. So there are a lot of outlines that coincide to the same, or are actually, you would have to describe to the same object. So you get an over complete set of polygon candidates that you then have to somehow disentangle. And this is a step which is called non-maximum suppression. And this calls for having a lot of candidates, finding out which candidates belong to the same object. And this is typically done by using the intersection of union criterion. So if it's two candidate polygons intersect above a certain threshold, then you just discard them and keep the one with the highest probability. And that's a very generic way to deal with this over complete set of bounding box or star-convex bounding shape representation, and it works quite well. Of course, the caveat is that this assumes intrinsically that your shapes can be represented by such an outline or by such a geometric primitive. And this is the case for what is called a star-convex object. And star-convex objects are objects where you can find a point in the center where you see essentially along a predefined set of, for instance, 32 rays, you see the boundary without crossing. So the green cells would be star-convex, whereas the red cells would not be star-convex. And this is just how the training process evolves. Here you see in the middle, you see the results, and to the left there's the input. In the middle, there's the training loss decreasing and the intermediate probability and distance calculations. And to the right, you see how the instances get more and more well separated as training progresses. And this, we applied that to a little toy model just to kind of highlight the strengths of and the failure cases of top-down approaches. And these are essentially like coffee bean shapes whose bounding box of neighboring 45-degree angled coffee beans are almost perfectly overlapping. So any top-down bounding box-based method, such as mask RCNN, should fail, and indeed it does, whereas Stardust and the bottom-up approach is quite well suited. In the case for crowded cells, then as I already showed you, the bottom-up approaches like the 2-class and the 3-class unit suffers, whereas mask RCNN, so a top-down approach, and Stardust behaves quite well. So this is an example on the Kaggle challenge from the Carpenter Lab. They provided like a training data set of nuclei and fluorescent labeled nuclei. And here again, you see, especially for challenging cases where mask RCNN and unit predict fusing cells, Stardust does quite a good job. And this can be quantified by the accuracy over the intersection over union threshold. And just to give you a short illustrative example, this works quite well even in very challenging cases. So this is an example from Anna Maria Zagorula in Manchester, where they had 10,000s of cells in a petri dish, mostly very decent SNR, but a lot of out of focus light. And you see it's very challenging to delineate different cell instances. But with some, I think 10 little crops of training data, it already did quite a good job in delineating the cells. And the same goes for this example of a whole slide image in histopathology, where the aim is the same. You want to find those nuclei and then do analysis on those nuclei in the same way. And as you can see, even on such large images, it does a pretty good job in delineating roundish nuclei. And of course, those nuclei are typically roundish, so they're well suited for our approach. The same works in 3D. just your distribution of rays has to first a little bit more well adapted, and secondly you have to have more rays than in 2D, because like a shape in 3D and essentially is a 2D shape, whereas in 2D it's a 1D shape. So you have essentially you need essentially way more points to discretize it perfectly, or to discretize it well enough. And you have the additional problem that you need that the data is typically anisotropic, so you have to adjust for that. But this all can be done. And an additional thing you have to consider is the polygon intersection for Polyhedra. In this case, it's a little bit more involved, but you can do that with a filtering process where you first describe bounding boxes, then you describe convex hulls, and you kind of filter those objects, and each of those individual progressing is getting progressively computationally more expensive. So you can do the easy steps first and only do the computationally expensive parts at the end. And this is just an example of a C. elegans where this works quite effectively too. What we have done for 2D so far is we provide some pre-trained models. So this is for fluorescence microscopy from the Data Science Bowl Challenge and for histopathology. This can be then pre-trained and distributed, for instance, via Fiji. So it's essentially a plugin where if you have images that are similar to one of the pre-trained models, you can just select the pre-trained model, and it even works on the CPU fast enough that you can just do it on your images in Fiji just by a click of a button. So what we recently did is two additions. The first one is object classification. So far only we're concerned with instance segmentation. But sometimes you have 2 different or 3 different cell classes that you want to additionally predict. And this can be done by just augmenting the Stardust network with an additional head, which predicts the class probabilities, and therefore have additionally class predictions towards additionally to the instance predictions. And the second thing is inference for large images. And what typically done is for very large images, you would tile those images, and each tile fits into GPU memory, and then you would kind of compute for every of those tiles the individual objects, and then do this non-maximum suppression step that I showed you before for the whole group of images. And this has 2 challenges. First, the runtime, and second, the memory. So the runtime, because you have to do NMS for all of those candidates, and memory because you need dense storage of n distances per pixel. And if you do it naive, so you just aggregate all of the different instances, and then you do or candidates, and then you do this non-maximum suppression, this disentangling on everything, and additionally predict dense instances, you get quite horrible runtimes and quite horrible memory. And so we thought of, okay, successively making it better, first with a KD tree that kind of implements a fast neighboring search algorithm that kind of can prune this list of neighbors that can be potentially intersect, can prune that quite down. So the runtime immediately goes down, whereas the memory stays the same. You can do sparse predictions. So you filter those candidates per tile and only stitch the filtered results, which gets down with the memory. And additionally, you can predict overlapping blocks, each individually do the NMS in each block and just take care of the intermediate ghost layer, so to speak, and make sure that they match. And with that, the memory condition comes down to almost constant memory. So you can predict 100K by 100K image in roughly 15 minutes, giving you 8 million instances. And if you're interested in, here are some places to start. Other projects that extend that or worth mentioning is Blindest von Mandal Ullmann and Virginie Ullmann in the UK. Super cool thing. And I want to just close with 2 cool projects that make use of that. First is Stardust as a first step in classification workflows, and this is for histopathology, where you implant little tumor cells of human and mouse tissue, and then you want to classify each individual cells from those histopathology images. And here you can use Stardust as a class agnostic nuclei detection, then extract cell features per the detected nuclei, and then train a classifier based on those shape features, and then apply it to reliably classify cells into human and mouse. And the second project, which I want to close, is a super cool project that we did together with Andreas Müller and Deborah Schmidt from Dresden, from the Solimene Lab, and Harold Hess. And this called for the reconstruction of beta cell organelles. And beta cells are those cells in your body that produce insulin. Insulin secretory granules, and they are implicated with microtubules, or microtubules are implicated in that, in the sense that those insulin secretory granules are transported, or are thought to be transported by microtubules and then being secreted. Those beta cells are part of the islet of Langerhans inside the pancreas, and of course the research question is, that's extremely interesting, is how does this microtubule network interact with secretory granules and other organelles, and how does this network change over time with stimulation of glucose? And Andreas set out to investigate that, and together with Harald Retz's group in Geneva, they acquired FIPSEM 4 nanometer quasi-isotropic stacks of beta cells in situ under 2 different glucose stimulations. And these stacks are extremely rich, extremely detailed. This is just an overview of the fly-through of one of those EM stacks. And you see like this little dark blobs are the insulin secretory granules. Others are microtubules. You see the Golgi apparatus, mitochondria, and it's a very rich source of data. And of course, you want to segment all those organelles, and then you want to correlate the geometric relations between different organelles of different types. Here's the nuclei. Very cool. And what we did, we essentially used a whole battery of segmentation methods that are adapted for each problem. So the different organelles, they pose different challenges in terms of segmentation ability, or the ability to being easy to segment or hard to segment. And we kind of used almost virtually a single different method for each of the different organelles. So for nucleus and centrioles, we could get away with semi-automatic, almost thresholding like a segmentation methods with the microscopy image browser. For the mitochondria, we used Elastic, super cool, easy to use software from the Crasher group and Heidelberg. Random forest. For the Golgi, we used the UNet based on CSP Deep with deep learning. The microtubules, we had to manually trace with web knossers, and by we, I mean mostly Andreas. So he had to trace all microtubules. And of course, there are some recent publications that aim to do that automatically, which would be so cool because, yes, all microtubules. And for the insulin secretory granules, we used StarDist in 3D because the roundish shapes of those secretory granules is well adapted to our approach. And this is just a short result. So these are individual secretory granules, of course, segmented automatically based on, I think, 6 crops in total of small cells that we manually annotated. And the output of StarDist is already like little meshes for each of those instances. So you can immediately put that, for instance, in Blender. You don't have to do any surface extraction from that. And with that, you can then actually build a comprehensive microtubule organelle interaction map for different glucose stimulation conditions. For 3 cells of the low glucose stimulation and 4 cells for the high glucose stimulation condition. And for instance, this is just, there's such a great richness of biological information there. And I just want to quickly highlight 3, that the microtubule length changes with glucose conditions and secretory granules are enriched near the membrane and the diameter is conserved. And finally, I just want to show you that it not only provides quantitative, extremely rich information, but just qualitative, very beautiful insight into the inner workings of a beta cell. And this is a video rendering of all the organelles by Deborah Schmidt, which I find so fascinating just to watch it all the time. And you see how packed the cells, how packed all organelles are packed inside of the beta cell. So everything that you see in light red are secretory granules, the C2 microtubules, and fly now through it. The mitochondria in blue, the Golgi in green, and the microtubule network with the centrioles. And of course, this could be done automatically or semi-automatically, and in the case of the microtubules, manually for 4 high glucose and 3 low glucose cells. If you're interested in, I urge you to read this publication on bioRxiv on Andreas Müller et al. And with that, I want to thank a lot of people that are mentioned here on this slide. I thank you for your attention and hope there's still some time for questions now or afterwards, but at least latest at the meet the speaker. Thank you very much.
Thanks, Martin. Amazing work, really.
Thank you.
Can I make a question as we have like 5 minutes or should we move on? No, I just I have the curiosity, when you are using the network for doing the restoration, how do you measure the quality of the restored worm?
So this is a super important part, that you always have to make sure that you validate your results accurately. And what we typically use is we have always like a holdout test set from a different animal and acquired at a different date, and then apply the network on those images that you have the ground truth for that and can easily see if there's anything that is unreliably detected, because that is an important, extremely important point, right? If you don't have that, for instance, just doing something pre-trained doesn't work there. That's just don't use pre-trained networks for this type of task, right? Because for segmentations, it's easy to see when a segmentation fails. But if restoration is a little bit more critical in that. So you always have to make sure that you have some test data set that you can validate it on. That's an extremely important point.
For the visualization at the end that Deborah created, do you know which tools?
This was with Blender.
Blender. Because they are amazing. As the most
they're super. Yeah, she's a brilliant. Yeah, she's a brilliant mind.
Okay, so thank you, Martin, again. So we would have all